So I think we should get started. So it's a great pleasure to introduce Dr. Stig Linder. Dr. Linder came last night all the way from Sweden and is going to spend with us a couple of days, like today and tomorrow. So he will have the chance to talk to many of you, and he has a quite busy schedule. Dr. Linder is a professor of molecular cancer pharmacology at the Karolinska Institute in Stockholm, Sweden. And more recently, as of 2016, he also became a professor of pharmacology at the Linköping University, which I understand is a lovely city a couple of hours south of Stockholm. And he's also a guest professor at the Tianjin Lung Cancer Institute in Sweden. Dr. Linder got his PhD in cell biology at the Karolinska Institute and has done postdoctoral training both in the US at Yale as well as at the University of Uppsala in Sweden. Dr. Linder has a very active research program. He has published over 160 papers and over 40 reviews and book chapters. And over his career, he has mentored and co-mentored over 20 PhD students, and I have had the pleasure to meet some of his students last year in his laboratory, and they are he's very much loved by them.
I didn't know that.
Well, now you do. Dr. Linder's research focuses on discovery, characterization, mechanism of action, and preclinical testing of small molecule inhibitors of proteasomal pathway and mitochondrial pathway for treatment of solid tumors. He holds 4 patents for anti-cancer drugs. They are in preclinical development, and I think today he's going to talk about one of the molecules he has developed called VLX1570, which is a molecule that is now in clinical trials in Sweden and in the United States. Dr. Linder serves as a board member of VLV Bio, which is a company that markets M30 apoptose, which is a kit to test apoptosis that Dr. Linder has developed several years ago. And he has also served as a chief scientific officer at Vivolux, which is the pharmaceutical company that has developed some of these inhibitors, including the VLX inhibitors that he's going to talk about today. So without any further ado, it is my pleasure to have Dr. Linder here, and we are all excited to hear about what you have to say. Thank you.
Thank you so much. Oh. So, thank you. Good morning. Does this work now? Okay, thank you. So, thank you for coming. I have a friend since many years that spent some years in Minnesota in the 80s, so say, enjoyed Stalingrad was his comment. And the reason that Swedes moved here that in the 1800s is that we are so masochistic. I don't know if that's true. But I didn't expect this much snow, to be quite honest. Okay, so I'm going to talk about one of our projects, which is related to the proteasome. And so our lab comes out of cancer drug research. So we've been studying chemotherapeutic agents for many years and their mechanism of action, including cisplatin. We spend a lot of time trying to understand how cisplatin works and other drugs. And the very quick summary of that is that these agents have very complex mechanisms of action, which is probably why they work on such a complex disease as cancer. You really need a multitude, a spectrum of different effects to get it, of actions to get effects on cancer. And having done that, our lab and a lot of other labs have gone into trying to use cell-based screening to look for interesting activities in drug libraries, to try to find things, drugs that have desirable activities, and then try to identify the targets and to validate the targets and bring these compounds further. The most popular way of identifying the targets are doing pull downs. We don't do that. What we do is a gene expression profile, and I come back to that. The advantage of this approach is that it's actually possible to discover new targets. and new druggable targets, you know in your project that you actually have a desirable activity. The problem is to try to figure out what does this compound do. And that has been a problem in this type of research for many years. And now we're getting better and better tools to do that. So life is sort of picking up. So we do a lot, which is a method that I actually recommend for anyone working in this field. We use a resource at the Broad Institute called the CMAP, the connectivity map. And this is a gene expression profile database. So what you do is that you treat a specific cell line, MCF7 breast cancer cells, 6 hours with an IC50 concentration of the drug, and you look at the response. What do the cells think that they have been hit with? This is not necessarily the same as pulling out the target, because there are many targets in cells. And at our institute, there have been some very embarrassing papers in Nature and other journals where people have a high-profile target, cells are dying, everything looks fantastic, and it turns out that they have a tubulin inhibitor. It happened twice. That you see using this technique, because tubulin inhibitors, which is about 50% of all drugs you would get out of a cytotoxic screen, they generate a very specific gene expression profile. And many other known pathways would do that. So that's a very good thing to do, to avoid the embarrassing of publishing a high-profile paper and then it turns out that everything is wrong. And people spend, reviews in Cancer Cells and telling you how stupid you are. It has happened. Okay. So we have 2 main projects that we're working on during recent years. One is the development of proteasome DUB inhibitors, the ubiquitinase inhibitors. And one is to discover compounds that are active on quiescent tumor cells that are present in hypoxic areas of tumors. And I won't talk about today, but the problem there is that there are, in hypoxic areas in solid tumors, tumor cell populations that are not proliferating. They will never be touched by conventional chemo and will always grow back. And that constitutes a major clinical problem. We've been screening steroids, small tumor clumps in large-scale steroid screening to pick up, and we had a compound that was quite successful. Won't talk about that. What I will talk about is a small molecule called BAP15. And that was identified in phenotypic screens. So we were looking for interesting molecules. One feature of this compound is that it induces apoptosis independently of p53. And that is what you would see here. This is an apoptosis assay that we developed. You can quantify apoptosis. You have lots of apoptosis up here. p53 wild type, p53 mutant knockout cells, the Vogelstein cell lines. Here you have compounds that induce as much apoptosis in the p53 plus as the p53 minus cells. Most cells are dependent on apoptosis, on p53 for apoptosis, but many are not. And this compound also induces apoptosis in cells that overexpress BCL2, which is quite remarkable. It shows robust cytotoxic activity in sub-micromolar range. It has a therapeutic window to tumor normal cells. We thought this was an interesting compound. Then it turned out that 2 other labs were working on similar compounds, one in Italy, in Udine. This is a lab headed by Claudio Brancolini. They published a paper in Cancer Research 2006, and they always draw their compounds in a way that's different to ours, but doesn't matter. They say that these compounds are deubiquitinase inhibitors. I won't go into, I'll come back to the ubiquitinases, but these are enzymes that take ubiquitin off target protein, substrate protein. What was a bit strange in this paper is that activity was observed at very high concentrations, 30, 50 micromolar, whereas the activity in cells, the accumulation of polyubiquitinated proteins was much lower. So we didn't quite understand this paper, and I don't think they understand our papers either, so that's mutual. But to this, we couldn't really reproduce these results. So another group was Martina Bazaru's group. They published a paper in 2011 showing things, very similar results that we have, compounds that have the same structure, similar structure, also inducing polyubiquitin accumulation in cells. And they found that this happens without inhibition of the 20S proteasome. I'll come back to the proteasome. We had the same results, no inhibition of the proteasome, still accumulation of polyubiquitin, and here are their results. So, what is a key feature of these molecules? Chemists hate them, okay, because they are potentially reactive. They are alpha-beta unsaturated carbonyls. We have a carbonyl group, and then we have the alpha-beta unsaturated alkene here. This beta carbon is potentially reactive to cysteine. So we have a reversible cysteine reactivity here. So we have two of those in our set of compounds and in the other compounds. There are a number of natural products that contain this reactivity. So curcumine, you see those here. And curcumine is quite a remarkable drug or compound because there are, I looked at yesterday on the plane, there are 4,357 PubMed hits on curcumine, and this is a compound that is not orally available and is not used for cancer treatment. And basically almost as many molecular mechanisms described. It's a very strange field. Hyperlongumine, pure, smaller inserts, also has this feature. There was a paper in Nature in 2010 showing that this is a very tumor cell selective drug. We have actually shown that it's a proteasome inhibitor, but that's outside the topic here. Anyway, we thought these compounds were interesting, so we did the CMAP analysis. We looked at the gene expression profile, how do the cells respond, and sure enough, they think that they have been hit by proteasome inhibitors. So the gene expression profile in the microarray analysis that is most closely related to the BAP15 signature is that of MG262, which is a very well-known proteasome inhibitor. So the cells think that they have been hit by a proteasome inhibitor. Okay, even if we don't see no inhibition of the proteasome. So that was a bit weird. Treating cells, we see accumulation of polyubiquitin in cells, which you would expect. That correlates very well with induction of apoptosis and accumulation of a number of proteins that are unstable in cells and known to be degraded by the proteasome. So this really looks like proteasome inhibition in the cell. So that looks good. This looks actually really good because what we see here is analysis using a reporter cell line. This cell line expresses an unstable ubiquitin YFP fusion protein. This is very rapidly degraded in cells normally. So untreated cells, you don't see any signal. If you block the proteasome, the cells will become yellow, okay, which you see here. Okay, fine. So how interesting is that? Well, we use a rather low concentration of the drug here, so not all cells get their proteasome blocked at this concentration. But it's only the yellow cells, the proteasome blocked cells that die, and they lyse, and the yellow YFP will leak out. But there's a perfect correlation between the pharmacodynamic effect of blocking the proteasome and the effect of killing the cell. I mean, if you would see that these cells will also die, then we would have closed the project because there's no correlation between cell death and proteasome inhibition. That's not true. So it really looks like we have a fairly clean proteasome inhibitor here. And that was good. This is interesting. because it's known that tumor cells are sensitive to proteasome inhibition. There are a number of different views on why. And my personal view is that tumor cells are more active in translation. They're more active. They make more protein. It's well known that a rather large fraction of newly synthesized proteins become misfolded and need to be degraded by the proteasome system. So if you block the proteasome system, that really hits tumor cells much more than normal cells. And one of the cells in the biological system that really makes a lot of protein is multiple myeloma cells. They make lots of immunoglobulins. They really need an active proteasome to take care of the misfolding. So there are three, I've listed two of them here, proteasome inhibitors that are approved by the FDA for treatment of multiple myeloma. So this is a validated target in cancer. We inhibit the proteasome. These compounds block the 20S proteasome. This is the 20S, which is the business end, which would degrade the substrate. And here we have the 19S regulatory subunit, to which the ubiquitin will bind and then be unfolded and the process be continued. Again, we had a similar result as Martina Bassaro's group, showing that DUB15 does not inhibit the 20S proteasome. This is bortezomib, one of the clinically approved proteasome inhibitors. It really kills proteasome activity. Our drug does not. These are 20S substrates, and there are some different substrates you can use. None of them is inhibited. None of these activities is inhibited by DUB15. So far, we seem to agree. Still, this was an enigma for us, but knowing that there's one report of the ubiquitinase inhibition by these type of compounds, and then the 20S inhibition, led us to think that, well, maybe the target are the ubiquitinases of the 19S, because there are three different enzymes in the 19S regulatory subunit which are needed for removing the ubiquitin chains of the substrates before degradation. The proteasome cannot, here you have the target protein, the polyubiquitin address tag that will target the substrate to the proteasome, will bind to receptors, but this cannot be degraded. You need to take off the ubiquitin. This will not fit into the 20S catalytic chamber. And there are three different enzymes, two cysteine proteases, isopeptidases, USP14, UCHLS, and one metalloprotease, POH1. They have different functions in this process. So it seems to be a rather reasonable hypothesis that these cysteine proteases would be targeted by the compound. We have a cysteine reactivity, DUB inhibition being sort of shown, and proteasome inhibition. And that turned out to be true. So these are different assays using different substrates, assaying DUB activity of the proteasome. I won't go into them. You have to believe me, these are conventional enzyme assays. This is an assay that is used a lot in the field, where you use a ubiquitin vinyl sulfone structure. This is a ubiquitin that will bind to active enzymes and modify it. And then you discover that detected by Western blotting. So a band here does not reflect the amount of protein. It reflects the amount of active, enzymatic active. And that is totally inhibited by our compounds, and it's also inhibited as expected by maleimide. So that was a good day in lab when we found this. There are not only bad days in the lab. The proteasome, if you do glycerol gradient centrifugation, you find that the activity is blocked at this stage. You will find the polyubiquitin on the proteasome. These are glycerol gradients, sedimentation this way, and here we have westerns for the proteasome subunit. polyubiquitin gets stuck on the proteasome there. There's a 5 there. I don't know how that came up. This compound BAP15 shows good activity in different both solid tumor models and in myeloma models. So these are both syngeneic and xenografts. There is a problem with these results, and that is that the compound is very insoluble. So we have to use cremophore for these usually IP injections and cremophore, and that's not good. That's clinically not good. That has been a problem in this project. But we get good activity. So BAP15 represents a novel class of inhibitors of proteasome. It does not inhibit the 20S as the well-known used inhibitors, but would inhibit the proteasome function at this stage. So that was interesting, but does that have any significance? Well, it would, because myeloma patients that are treated with proteasomal or other 20S inhibitors ultimately develop resistance. They fail therapy. And we would expect, due to the different mechanisms of action, that these compounds would work on proteasomal-resistant cells, and they do. So we have shown that for our cells, and it's also shown by Ken Anderson's lab at the Dana-Farber and by Asher Shalan Khan at the Mayo Clinic, that proteasomal-resistant cells are sensitive to BAP15. So that's good. So we have a possible future therapeutic indication for these type of compounds. I mean, if it's our compound to someone else, but the target is a really good one, in our opinion. Very important for work in this field is to validate the target, and that is not always done, and that also can become quite embarrassing. We have done knockdowns. A little bit tricky because the cells are dying at the same time. But the knockdown of USP14 UCHLS does result in loss of viability, relative cell number here, and not so much with a single sRNAs, but particularly with the double, inhibiting both the enzymes to get the accumulation of polyubiquitin. To study this in more detail, we have developed, and this is Dig Darcy in the lab, who's done a lot of this work, develop a conditional knockout of USP14, where we have floxed exon 5 in colon cancer cells, and when we infect with a Cree virus and put the cells into a cell sorter, we can actually follow what happens with deleting USP14. And this is an IncuCyte experiment. You have time here, and number of cells, and these are the control cells, and this is deleting USP14. The cells look really strange, get very large nuclei, and we have a lot of cell death in here. We don't quite understand what's going on. It's very interesting to try to figure that out. But we can validate the target, and that, if you don't do that, it can also be quite embarrassing. So we have then tried to lead optimize BAP15. And we did that together with a company that synthesized around, I think it was 180 different compounds. We came up with this one, which is not perfect. We kept the acrylamide because it didn't really make a difference, and it in terms of the profiles and everything. And so this is what we have been going with. Not perfect because we still require we have some solubility problems, still require some cremophore, which is not perfect. I come back to that. VLX1570 preferentially, and this is true also of BAP15, preferentially inhibits USP14. So here you have these activity plots again, where a band represents enzyme activity, not amount of protein. You see that USP14 is preferentially inhibited here over UCHL5. And we can see binding to the target using BioCore, surface plasmon resonance. Actually, it's a technique developed in Linköping, where I work during the weeks. So USP14 we have a lower KD to USP14 than to UCHL5. This is a suspiciously high KD. The problem with interpreting these results is that we're not quite sure of the intracellular drug concentration always, but we have a preferential uptake of the drug. So the drug concentrations in the cell can be quite high. But it looks reasonable. This compound, this is of course our best experiment ever. But in this particular experiment, we cured the mice. This is an orthotopic model of myeloma. And the mice obtained treatment after 1 week and for 1 week after that, every day. But it really made a big difference. So all the treated mice survived. This is not always true. This experiment, and we didn't do this experiment. This was done by a company, so it made it even more comfortable. A major problem with this project and with these molecules is the general reactivity and possible off-target activity of the compounds. I mean, okay, we have cysteine reactivity. There are a lot of cysteine cells, although most of them are buried inside proteins. There are not so many proteins, cysteines on the surface, but still, this is a problem. So there are about 80 different deubiquitinase enzymes in cells, and there are about 50 of those are USPs. So similarity to USP14. So does it make any sense to claim that we have selectivity for USP14? So, of course, a major problem or a major question in a project like this, working with a drug like this. And in our hands, we don't see a major off-target. We don't quite understand this because it doesn't make much sense, but you can only do the experiments and present them. We don't see, this is a total cell extract experiment. BAP15 does not inhibit. This is an experiment where this is 1670 actually. We sent that for profiling at the company. Don't really see much. This is a nice type of experiment because it then make you sort of confident. Here, these are cells treated with the drug. And the loss of the band here means loss of activity. This is inhibition. But in the treated cells, if you then use other antibodies, this is USP14 being inhibited. UCHL5 a little bit, but USP7, UCHL3, and all the others that we have looked at are not inhibited. And these are the same cell extract, the same treated cell. So we don't see much off-target activity. And Dr. Bassaro published a paper in Clinical Cancer Research 4 years ago, essentially showing very similar type of data. I mean, proteasome, DUB inhibition, not general DUB inhibition, by a compound that you call RA9, which is very similar to BAP15. So we agree. The Italian group does not agree. They do pull-outs. This is a biotinylated compound, similar to ours, and they pull out all sorts of DUBs. And I honestly don't understand the difference. I think conservatively, if someone shows this, I think this is actually what you should believe. I mean, nothing else has been proved. Still, the pharmacodynamic, the molecular mechanism of action that we see with these compounds is very rapid cell death. Proteasome inhibition, a gene expression profile that is typical of proteasome inhibitors. So in my mind, even if we inhibit other DUBs, maybe that doesn't make so much of a difference because, I mean, you have this rock concert going on the proteasome, and then someone is playing a violin at USP18, and who cares in that situation? I don't know, but you can argue against that. I can probably myself argue against that. But anyway, that's where we are. It would be strange if these compounds do not have other targets. I mean, I can only present what we find in our lab. And we really looked hard because, of course, this is very important for a project. Popular compounds. This is a paper that came in Cancer Cell 5 years ago from Richard Roden's lab, which is your previous PhD postdoc supervisor. They have a compound also very soon, start to recognize this structure soon. They publish that this binds also to the proteasome, but not to USP14, but to a ubiquitin receptor called RPM13. The little bit confusing result here is that it doesn't bind to the ubiquitin receptor part that would actually recognize ubiquitin, but it binds on the other side here. And they have a theory for how this works, which, to be quite honest, I don't quite understand it. I don't know how many times I read those sentences, but all I can say that we do not find inhibition of ubiquitin or polyubiquitin binding to proteasomes. So that happens. So we don't see inhibition of the receptor, and that would make sense from what they say too. This does not. We can do these glycerol gradients under conditions where the USP14 falls off. If you increase the salt a little bit, USP14 is just loosely associated. So the polyubiquitin is bound to the receptors. It does not get stuck to USP14 due to the catalytic or whatever block here. So one possibility to sort of merge these results is that it's known that RPM13 actually will bind and activate UCHL5, which is also called UCH37. So maybe the UCHL5 inhibitory activity that we see is due to these compounds also binding to this protein, to this receptor. I don't know. So that's where we are. I think I'm doing reasonably with time. Okay? Don't look so worried. Yeah. Yes. Yes, absolutely. Resistance. Very good point. So this has been a problem in that it's been very difficult to generate resistant cells. We can generate cells that are about twofold resistant. And there's nothing wrong with the target, probably because you cannot really mutate this target. The cells will die. What these cells have is a somewhat higher glutathione content, so that they actually inactivate the compound. But it does not give us any clue of the target, which is a good thing from a clinical perspective. You don't want resistance to develop in, after 2 treatments. Resistance always, it's ridiculous to say that you don't get resistance to a cancer drug, you always will. But in this case, it's not so pronounced. But it doesn't tell us much about the target.
It would help optimize, though.
What?
It would help optimize.
Yeah.
If you knew structurally where you're going.
Yeah. All right. So, having done that, working with these molecules, there were 2 major things that have been worrying us. One thing is that the solubility has been a problem. We have this long conjugated system, and whatever we do, it's really hard to get good solubility. It's hard to get good pharmacokinetics. We have had a problem with that. We have a pro-drug agenda, which looks rather promising for the 1570, which I can't talk about because the company would kill me. But that has been difficult. The other question is the general cytotoxicity, the general reactivity of these compounds. So we have the Italian group that say that they inhibit everything, bind to everything, and then we have our data that say that they don't. So who is right? How cytotoxic are these alpha-beta unsaturated carbonyls, also called enones? Some people really hate them. They call culprits, and the drug discovery, and they should be really avoided by the companies. The pharmaceutical companies really hate them. The chemists really hate them. They have changed a little bit because there have been, during recent years, several very successful tyrosine kinase inhibitors that are irreversible inhibitors, made it very quickly to the clinic. So companies are changing their mind. Anyway, what we finally decided to do to look into this, to try to get an answer with regard to the reactivity, is that we contacted Cambridge, it's the vendor, and we wanted them to pick out all the alpha-beta unsaturated ketones that they had with a reasonable log P. This is solubility, and this would be a good range with what, because these compounds would be membrane permeable, they would not be incredibly insoluble, so that would be reasonable. And we took those compounds and we screened them for blocking of proteasome function in the cell-based assay based on the ubiquitin rhodamine, ubiquitin YFP. We started with almost 5,000 compounds, and one horrible possibility here is that it would all be toxic, and we would just close down the project, and that was not the case. So at 5 micromolar, only 3% of these electrophiles were toxic, which is a very surprising result. After some validation here, 36 of those blocked the proteasome. A surprising result that so many of the cytotoxic compounds would be proteasome inhibitors. Probably if we had used another concentration, 25 micromolar, I'm quite sure that this would have looked different. I have many more cytotoxic compounds, but this is what we see here. We picked our 10 best hits and tried to look at what do these compounds do. And they are here. 3 of them have the same pharmacophore that we sort of have working with. And 7 look completely different. I mean, they all have these in the screen, so only screen these compounds. These compounds all block the proteasome, polyubiquitin on the proteasome, so the substrate gets stuck on the proteasome, and they inhibit 19S deubiquitin, not 20S proteasome activity. Basically, all of them inhibit 19S deubiquitin. And they are not also show reasonable selectivity. These are total DUB activity, generally not inhibited. 2 different assays, chromogenic assay and the ubiquitin vinyl sulfone assay. And they seem to get selectivity from these compounds, all of them, which, I don't know, if someone would have told me, I wouldn't have believed them, but this is what we find. Since we have so many compounds, we can start to ask questions about the properties of these electrophiles. So one possibility would be that the cytotoxic compounds will have a much larger reactivity. You can calculate the electrophilic index, and that was a little bit higher, but not such a large difference. So this is the degree of plus charge on this carbon that would react with the system. And the electrophilicity did not correlate to the IC50, to the cytotoxicity of the compound. Also surprising. Then we have some really good friends at the Karolinska that work with an enzyme called pyridoxine reductase, which is an anti-oxidative defense protein. This protein contains a very highly nucleophilic selenocysteine. It's the most nucleophilic, probably, enzyme or cysteine in the cell, and is known to react to electrophiles. So we thought that possibly all these 142 cytotoxic compounds would all inhibit TRXR. Not true. Only 20 of 142. There is an overlap, but the majority of our UPS inhibitors do not inhibit the TRXR. Very surprising. I mean, Elias Aner was the TRXR expert. He was astounded by this, and also very disappointed because he thinks that TRXR is sort of the center of the universe, as we all do with our targets. Another thing we can do here when we have so many compounds is we can do docking. And it turns out that we can dock all of them very close to the catalytic triad of USP14. So here is USP14, catalytic triad. Here is ubiquitin binding in. And there is a groove here. That's the only druggable site that you see in USP14 using PMOL in these programs. And all 10 block here and are expected to block ubiquitin binding to the enzyme, which was also another good day when we found this. So that's really nice to see. You can argue against molecular docking, but when you have 10 compounds that all do it, then the evidence starts to get stronger. We have also looked at, not doing fantastic with time, so I'm going to go through this rather quickly. These are binding experiments. We can, with different methods, show that all these 10 compounds bind the enzyme. This is intrinsic tryptophan fluorescence. It worked very well because there are some tryptophans very close to the catalytic triad. Now, what we would expect here is not only binding. We would expect reactivity with the cysteine and getting irreversibility. So are these inhibitors irreversible? And the answer to that is yes, but not always. So we have one compound here, 360, that does not seem to be irreversible. So what are these bars? This is a dilution recovery experiment. So we treat the 19S proteasome, 37 degrees, with 1 micromolar, 20 micromolar, 20 micromolar followed by dilution. And if we have a reversible inhibitor, the dilution activity would be the same as 1 micromolar all the time. And that is true here. So you see 1 micromolar and 1 to the dilution is the same. For many of these others, that's not true. So we have both reversible, surprising, irreversibly, not very surprising at all. But they will all bind to the pockets, and of course you will get a better effect of the compound if they actually will bind to the cysteine. We can, for some of the compounds, this is a problem for us. I will just send this paper in for review, and the reviewer will probably kill us for this, but this is, you have to report what you see. These are moldy mass spec experiments. You see the change in molecular mass. Except for these 360, which was the reversible inhibitors, we see a change in molecular mass, not perfectly corresponding to the expected molecular mass. Though. That's what we see. But we don't quite understand that. But we see evidence for many of the compounds of irreversible inhibition, irreversible binding. And the model would be, we have a rather low KD here, or high KD, I mean, not that incredibly strong interaction with the enzyme. The docking scores are, except for one of the compounds, is not fantastic, but the compounds would sort of bounce in and off this pocket, and sometimes they will get interaction with the cysteine, so they stick there, and then we really get a strong effect on the enzyme and killing of the cell. We've done gene expression profiles, and these are really hard to explain, but this is CMAP again. What this basically says is that all these 10 compounds, again surprising, they induce an expression pattern that is typical of proteasome inhibitors. So not only do we inhibit the proteasome, accumulate polyubiquitin, but the expression profiles, chaperone induction, oxidative stress induction, etc. It's very similar to that. So we have a number of compounds then that would serve as leads for further development. And okay, maybe we're sending in the paper, the review will say we have to test all these compounds in mice, and I think that's unreasonable. So what we have done is that we tested them in zebrafish animals, where you inject labeled cells into zebrafish, and you see if you get increase in cell growth when you add the drug to the water. I mean, basically in the aquarium. And we get inhibition of tumor growth. I don't think it's right. Tumor cell growth would be better because we don't have many cells here that we use. Get also inhibition of metastases. You can see how these cells spread in the got that picture. It's one here. You can see it's a spread to the tail of the fish or not. And we can see that actually inhibit the proteasome. You get UBYFP activity in vivo by adding the compound to the water. Okay, so to finish off here, it's surprising that we see this degree of selectivity. At least we see that. The USP domain proteins have similar structures. And this is, I mean, if we can, DUBs are quite interesting targets. You can really get to interesting proteins indirectly by affecting their stability via the UPS. So like the P53 would be a great target. And there are a lot of efforts on that. Here was a paper published last year in Nature, quite a crowd of workers here. They describe a USP7 inhibitor. USP7 is involved in the stability of HDM2, regulating stability of HDM2 and P53. So inhibiting USP7 would lead to a stabilization of P53. So this is a pretty good target. They showed a compound, or two compounds, that inhibit USP7, binding to exactly the same type of pocket that we see binding to with our compound. So here is our compound. You see the catalytic triad, and here are the binding pockets. They don't see USP14 inhibition. We don't see USP7 inhibition. But we found that there is a difference. There is a loop on top of this cleft where you actually see a difference between USP7 and USP14. So it's possible, I mean, that the dynamics of the DUBs and the possibility to get compounds in there is much larger than we would have thought. And that would be actually pretty good, because these are really good targets to go for. And if it now turns out by these different studies that it's possible to really get more selective DUB inhibitors, I think that would be a really good way to go for this field. So finally, conclusion. I think that, and this is no surprise to anyone, but you can use this type of approach to identify new targets. And we think that these alpha and beta unsaturated carbonyls found in curcumine and various natural compounds are less generally reactive than generally thought, especially by chemists. And one should not maybe just this is really throwing the baby out with the bathwater. You have to do the experiments and not only say that these are trapped compounds. So we believe that, curcumine, piperlongumine, many of the effects have been, multitude of effects been shown by these compounds. They are incredibly popular. And we think that many of them are probably due to effects on the proteasome system. We've shown that for piperlongumine, actually published that. No one ever cites that, but anyway, it's true. And I think it's reasonable to think that DUBs are more selectively druggable than previously thought. And that's really nice. It's known, and I think I just want to make that message again, that inhibit that going for ubiquitin, for the ubiquitin proteasome system, it's a good way to try to drug targets that otherwise are considered undruggable. There's an industry of people trying to drug transcription factors, for example. And if you look at the number of binding sites there, etc., that's really, really hard. Many of them do that by just screen for 60 compounds and they find something. If you do it indirectly via the UPS, I think you have a much larger chance, because the UPS contains really some very druggable enzymes. And I think that, we are very fond of our targets, the proteasome DUBs. I think that would be a good way to go. We're trying to either develop the VLX1570 and the compounds around that, prodrugs. or some of the new compounds that could be lead of time. So these are people. So I have one laboratory in Linköping, and Padrig is an Irish guy, very fond of Guinness, and here are people at the Karolinska, and we work closely with a very good structural biologist, Maria Sunnerhagen, who actually her name is there but not there. Okay, so it's Maria Sunnerhagen, and some people at Karolinska, and we also collaborated with Mayo and Dana Farber, the data that are shown. So thank you so much for listening.
So I think we have a little bit of time for questions. Any questions for us? James.
Hi, nice talk. Thank you for listening. I have two questions. One of them you might have said, and I missed. The other one I think you didn't. So you made a knockout, I think, in the colon cancer cells, getting rid of one of the subunits of the S19 subunit, right? You made
USP14, yes.
So my first question is that knockout now resistant to the, because the cells still grew. So are they resistant to the activity of BAP15 and all these other compounds? My first question. My second question really quickly is, the cell activity is remarkable, and I wonder how do you think you can actually give this to animals without wiping out their bone? Because the misfolded protein response in the bone marrow needs to be really hyper. So sorry, two different questions.
Yes. We have a lower. The first question is really something interesting a lot with regard to the knockout cells. They have a lower sensitivity. We still see an effect there. We think that there's another. We still get proteasome inhibition. We don't see how the cell. I mean, we think we have a compensatory effect of another DUB going there. So that is really a very active, which I don't have a really good answer to this. I'm really working on it, but sensitivity really decreased. But there's something weird going on there. With regard to the bone marrow, I think that's a general question for proteasome inhibitors, isn't it? I mean, why would we see worse effect by 19S inhibitors than 20S inhibitors? I think it's remarkable that proteasome inhibitors have a therapeutic window, but they do. And I mean, people didn't expect that. I mean, proteasomal wasn't really found originally. I mean, people didn't look for an anti-cancer drug. It was a multiple myeloma patient responded. And what we see with that is that, I mean, if you take a normal cell, like an immortalized fibroblast, they don't respond very well. If you treat those with a drug that inhibits ER translocation, so you induce a lot of more cytosolic misfolded proteins, then they become very sensitive. I really think it's an issue of the number of substrates that are there.
Wonderful talk. I have two related questions. So one was that, so if you reduce glutathione levels in the cells, do they actually become more sensitive to the inhibition? And the second question is, a lot of age-related neurodegenerative disease and heart disease are associated with reduction in glutathione levels and increase in oxidation status of the cell. So in that case, would you see more general, and also you would also generate a lot of carbonyl, alpha, beta, unsaturated carbon. So would you predict that in those conditions you would have a general inhibition of ubiquitin proteases under those conditions?
Second question, I don't think I can answer. Okay, so with the first one, we look at a lot of glutathione, obviously. So first of all, we don't deplete glutathione. There are some drugs out there that are example that are really cysteine reactive, and they will wipe out glutathione and you get a very strong oxidative response. We don't get that. If we use DSO to deplete glutathione, we don't get that much of a response of the colon cancer cells or myeloma cells. The cells that develop resistance, that is reversed by DSO. But for some reason, we don't see so much on the parental cells. That's the reason. I don't quite understand why, but that's our data. This is a general question of many reactive cancer drugs. I mean, like cisplatin, glutathione, and many others would be resistant factors, and they still work for sometimes strange reasons. These are not, from where I'm coming from, I mean, if you were working for headache or something, and some other pharmaceutical areas, you would hate these compounds. If you work with if you work with cancer, you really don't care. I mean, you want compounds that kill your cells quickly, and if they are target activities, they shouldn't. But if they kill the cells, I think that you're fine.
I think that sometimes some people, I've heard companies, people say the dirtier the better. In that you can have multiple DUBs, for example, or multiple DUBs activity being upregulated at the same time, particularly in a cell that is proteasome resistant, which I think it's similar to question, but then I have a question, which is something I never understood. I've been working in this for a long time, and I never quite understood.
Kind of around the same idea here. It seems like the tyrosine kinase inhibitors, they're going back to less specific is better.
Yeah.
So why are you so laser oriented on getting a specific inhibitor, especially if your goal is to inhibit proteasome? Why not go the opposite way and try and find a multi-DUB inhibitor? And along with that, I assume you've tried the combination of bortezomib and ubiquitin together. Would you see that as maybe beneficial, especially for, say, a multiple myeloma patient who you think is obviously going to mess up?
You see that you have done that, so you see synergy after effects in vitro. We have never done animal experiments. Pharmacology becomes very complex. I mean, that's true, but that question has two sides. I mean, fine, as long as they work, you don't care. But it also becomes it's an academic question. I mean, you want to know, and these are strange results. You want to understand it more at that level. Drug companies might not necessarily agree with you. I mean, they would if you want to sell the product, it's too reminiscent of very general chemotherapy. I think that the drug companies get worried, and then you have a problem with developing your compound. But in many respects, I agree with you.
I have a question related to the many of these DUB inhibitors can kill cells that are resistant to proteasome inhibitors, whether they are coming from patients that have acquired clinical resistance to it or whether they are done in vitro. And I always wondered, why is that? In the sense, how can a cell survive and thrive in presence of proteasome inhibitors? How can a cell work without the 20S working?
Okay, so why do cells become resistant to 20S?
Right. How can they survive if it is true that a cell that is adapted or
yeah. There are a number of papers on that. I think so. One mechanism is the beta-5 mutations, which will not affect the DUBs. Another is changing subunits in unipersonal subunits. That's one way. BCL2 overexpression, which these compounds are insensitive. I think there are a number of mechanisms where they really differ.
Do you think that those cells upregulate difficulty? Do you think that cells that are resistant to proteasome inhibitors are upregulating that?
We looked at that. We haven't seen that.
Any other questions for Dr. Linder? No. Thank you so much.